Nanomedicine Innovation Center that I direct. And this center, at this center, we have a strong program in development of materials for medicine. And we have, over the last 2 decades, synthesized more than 50 nanoplatforms of various kinds, some of which are shown here. Just 1 second, please. I'm going to turn on my clock here so that I know the time correctly. Yeah. And these materials were developed for delivery of a variety of biological agents, localized to amplify energy delivery, radio-sensitization, and for multimodal imaging. And some of the platforms are briefly described here. We've been fortunate to be funded by a variety of agencies, particularly the NIH, the Department of Defense, the National Science Foundation, and various private foundations also. Now, we have a very strong education program here, which is actually quite unique. We are one of the world's first nanomedicine certificate programs. We have developed courses over the last 15 years with NSF funding, and we also run a training program called CanCure. Now, in addition, I've also spun off 2 companies that were developed to translate the technologies from my lab into the clinic. And briefly, let me show, direct you to some of the websites. This website is my own, and it tells you, about all my various activities. The Nanomedicine Innovation Center website is here, and it tells you about all the activities at the Nanomedicine Innovation Center since its founding over the last 15 years. One of the programs, the training programs that we run is called CanCure, as I mentioned, and this is in collaboration with Dana-Farber Harvard Cancer Center. And you can see more about this program at this website. And then, as I mentioned, strong nanomedicine educational program. This is run under the auspices of the Nanomedicine Academy that I direct. And you can find out more about this at this website. So, so coming now to the kind of research that we have been doing, there is actually, as you can see here, there's more than 100 papers or so on these various topics. I'm going to focus mainly on 2 areas. And the first is, and so first let me give you an overview of the research themes of the center. One of the main areas is drug delivery. And while we have done various types of nanoparticles, as I mentioned before, I'm going to focus on nanoformulations using PARP inhibitors. We have a strong program in nanotechnology for radiation oncology, arising from my appointment at Harvard Medical School. And we have developed implants for radiotherapy, for radio-sensitization. And in fact, one of these is now almost, we're doing the IND enabling studies to get FDA approval. And we hope to be in the clinic in 2022 or 23. We have several articles on gold nanoparticles for amplifying radiotherapy. We've done an extensive amount of work on magnetic nanoplatforms. And I will here talk about one particular aspect of it where we have made what we believe is a breakthrough, which is magnetic MRI contrast enhancement, where we have developed a new modality of vascular imaging. And then the other aspect of all our work, as I mentioned before, is our training programs. So I'm going to start with the nanoformulations for PARP inhibitors. So PARP inhibitors are potent therapeutics that have come into prominence in the last few years. There are numerous clinical trials ongoing, but there have also been various approved nanoparticle, not nanoformulations, just the free drug formulations of PARP inhibitors, olaparib, which is AstraZeneca, BMN 673, which is now called Talzena or talazoparib, is owned by Pfizer. Rucaparib and veliparib are others that have also been approved for specific indications by the FDA. So the key aspect of how the PARP inhibitors work is based on a concept of synthetic lethality. And the concept synthetic lethality. The concept of synthetic lethality is that if you have a DNA break, a break in the DNA, the cell tries to repair the break, and it recruits an enzyme called PARP, or ADP Ribose, in a normal cell, those DNA breaks are repaired so that they don't amplify and essentially lead to cell death. Now, as a consequence, when you have a DNA break, you have an upregulation of PARP. Now, if you block the PARP inhibition with the PARP generation with an inhibitor, then of course you can prevent the repair. But if the cell is a healthy cell, it turns out that there are other mechanisms that it can utilize to repair the cell, and so it is still intact. So just inhibiting with PARP alone to a healthy cell is not going to make any difference. However, if it has one of the mechanisms, such as a BRCA mutation, then it turns out that the combination of the mutation and the inhibition of PARP leads to a cell death. There is no way for the cell to recover. And so this is really the whole point of exploiting PARP inhibitor nanotherapy, particularly for BRCA mutations. And this is this is done in a variety of settings. And as you may know, BRCA mutations are important in ovarian cancer and breast cancer, and there are also other defects in homologous repair and non-homologous end joining in other cancers also. Often the PARP inhibitors are used with something DNA damaging agents like cisplatin or radiation. So what we have done is, if you look at the current drugs which are given orally, they have very poor bioavailability and pure tumor accumulation, and so we have developed nanoparticle formulation which are injected into the tumor. They modify the half-life of the and extend it to from almost up to 24 to 48 hours, which then makes them long circulating and enables them to accumulate in the tumors. And so that's the advantage of the nanoformulations. We have carried out a whole range of studies in several cancers, including ovarian, prostate, breast cancer, Ewing sarcoma, and lung cancer. And there is numerous papers that have been published on this topic. and in fact, there are more that are coming out. And you can see, our details of our work in all these papers. We have done both in vitro and in vivo work using various animal models. So let me give you a quick summary of some of the results of our work. In addition to the nanoformulations, we also, so the types of nanoformulations we have developed are for IV injection, and these are nanoparticles of olaparib or talazoparib. This is actually nanotalazoparib is our leading candidate right now, and they have diameters under 100 nanometers, and they have a very long release time in vitro. Excuse me. We've also developed a sustained delivery implant, which is placed into the tumor and releases the drug over many days, typically, up to 30 to 60 days. And so the results of that implant are shown here. You can see here, and this is in a BRCA1 mutant model developed by Karen Leiby at Michigan State, who has been our collaborator on this project. And you can see that for the one with the spacer implanted in the tumor was essentially arrested over at least more than 30 days, and survival was greatly enhanced. Now, one of the problems is you do have recurrence, and that can be taken care of by adding more, inserting another spacer, or in combination with other drugs. We've also done work on using the nanoformulation injected into the BRCA1 mice, and you can see that the nanoformulation outperformed the free drug formulation and led to much longer survival. We have also now we have carried out extensive studies in ovarian cancer in a similar BRCA1 model, and the key message of that is that the nanoformulation controlled the time to ascites formation, and you can see that here the control, develops ascites for which they have to be sacrificed very early. The drug given orally, you can see tremendous toxicity. But with the nano drug, you can see the mouse is pretty healthy, and the ascites are also controlled. We've done extensive work in sarcoma, and I should add that all of these are published in papers, and you can see our sarcoma work is summarized in this paper here. And here we're testing it in combination with temozolomide, and you can see that in the combination drug gives you remarkable suppression of tumor growth. And you can see more details in this paper that's described here. We did an extensive study of nano-olaparib in combination with radiation, and that is described in this paper here. The basic message that I want to point out is this synergizes very well with the radiation, and the mice that were treated with nano-olaparib and 10 gray of radiation, you can see had the best survival statistics out to at least 12 weeks. So that summarizes our work in using formulations of PARP inhibitors for treating a variety of cancers, particularly the BRCA mutant cancers in breast and ovarian. But this is our formulations are also very efficient when combined with other drugs like temozolomide, and also we have worked with another nanoformulation of a CDK12 inhibitor, nanodinosiclib, that we have tested in breast cancer. And we've also done work in lung cancer. All of this work is described in the papers that I cited before. We're now, by the way, taking these formulations and we're looking to translate them, with under my company, Theranano. And I'll mention that briefly later on. So I now want to turn to my other topic, which is MRI using nanoparticles for magnetic resonance imaging contrast enhancement. And my key collaborators here are described here. I will also summarize all my collaborators at the end of the talk. So the idea behind this work is that the magnetic nanoparticles lead to what is known as the shortening of the T1 time. And that means that you get a strong signal at very short times, which you can then exploit. And that signal is amplified over the background. So wherever there is magnetic nanoparticles, that area, the MRI signal is modified in that area. So this is the whole basis of contrast enhancement. The traditional contrast enhancement agent is gadolinium, which is used extensively. However, it is now known, so by itself it is toxic. It is chelated to avoid the toxicity and designed to be excreted out quickly. However, there still is known that it is retained in the body, and that is, causing a lot of concern, particularly for chronic kidney disease patients who can be affected, who can then get a disease called NSF, which is extremely nasty and dangerous. So there's a search for an alternative contrast agent, and magnetic nanoparticles have been known to be such potential agents. However, the previous sequences, the way it was used in the MRI, it was used doing what is known as T2 weighted, leading to negative contrast. And you can see here that where there is magnetic nanoparticles with increasing concentration, you get a dark signal, which is not very good in the clinic. So radiologists don't really like to use it. What we did was we developed a separate sequence in which it turns out it becomes bright. And so now wherever there is blood and where there is this magnetic nanoparticle, it turns bright. And this is positive contrast. And this is of great interest in the clinic. So I will show you so some of the first work we did, and this is the first paper is described in this article here. We showed in mice that you can get exceptional clarity and definition in imaging the blood vessels. So that's one thing. The next part is that it's also quantitative, so it can be an assay, absolute assay of the concentration in the blood pool. And so that turns this into not just a camera, but actually a quantitative instrument of the blood volume. And thirdly, this is using ferromoxetol, which is much safer than gadolinium. And we did a very detailed study in rat brains, and this paper was published in Neuroimage. This work, we obtained entirely new angiograms of the rat brain, and also we quantified the blood volume in different parts of the rat brain to show, where's the blood flow. And really, the function of the brain is determined by blood flow and oxygenation. And so we're now able to quantify the regions where blood is present and where it's deficient. And that could be a diagnostic for a variety of cerebrovascular diseases. Now, it turns out that the agent we've been using is also approved for humans. However, it is not approved for imaging, but for iron anemia. And we can use it in humans for MRI with approval of the Institutional Review Board, or IRB. And so that's what we did, and this is work done at NGH under an NIH grant given to my company, Theranano. People with chronic kidney disease cannot be given gadolinium. So they cannot basically be imaged, but with this technique, they can be imaged safely. And that's what we have done. And this paper is now going to appear, has been accepted for publication in Abdominal Radiology. And what we can show here is we can see the kidney vasculature, we can see the vein and the arteries, and you can see the cross sections very clearly. You can also see stenosis, and you can see cysts, as you can show here. So this is a very important development both for brain imaging and for kidney imaging. Now, before I stop, I want to mention that we're transitioning these technologies to the clinic, and this is done through my company Theranano. The QUTE-CE is already in phase 1 clinical trial, as I showed you. There's a slow release implant of docetaxel that is now, we're doing the IND enabling studies, and we hope to get IND approval this year or early next year and start a clinical trial next year. And then we're also working to translate the NanoTalazoparib formulations. And we hope to be in the clinic very soon on that also. So to summarize, I've shown you really only two. We have a large program, but I've only shown you two parts of it. One is the drug delivery program, and then the other is the MRI program. This is only a short description of some of the people who have been working with me over the years. There's actually, the whole list is much, much bigger than this. I also want to thank all my sponsors who have supported me at NIH, NSF, DOD, the American Lung Association, the Rivkin Foundation, and the various hospitals that we have collaborated with. Thank you very much.